Hi class, it's me, your favorite teacher, best 10th grade biology teacher in Yellow Medicine East High School, Mr. Suter. I bet you were thinking you're going to get a day away from having to listen to my voice, but I thought it might be important that you guys follow the instructions with this lab, and I thought I'd give you guys a step-by-step show of how you're supposed to do things. Alright, the first thing we are going to start with is a piece of fruit. And today I chose a banana, even though your directions say that you are going to be using a kiwi, and then there's a typo even at another point where it says strawberry. We are going to use the banana. And we don't need a really big piece of banana, just about a chunk this size. And, you know, one of the key things that we have to realize here that fruit is also a living thing just like us, and it has DNA just like we do. And sometimes people forget that. In fact, 60% of this banana's DNA is exactly the same as our own. First thing we're going to do, we're going to put the banana inside the Ziploc bag. Then, we're going to take our detergent. This is just some shampoo mixed with distilled water. And we are going to take 20 milliliters worth of that, and you're going to measure that in a graduated cylinder. And again, pour the detergent or the shampoo water, 20 milliliters worth. And you're going to also place that in the Ziploc bag. Okay? Now, the part that's going to keep you from getting dirty today is, number one, you want to make sure you get all the air out of the bag. You don't want to lay it down flat. Try to keep all the content inside your bag, but get the air out. And then we want to make sure we get the clear seal in the bag. Yellow and blue make green. Now at this point we're ready to go on to the next step. We're going to smash this banana up. And between the smashing action, smushing with our fingers, and the detergent, we are breaking down the cell membrane and also the nuclear membrane that holds all the genetic material inside the cell. So what we're going to end up with here after we mix and mash here, is a shampooed solution mixed up, hopefully with some DNA. After you've got it good and smashed for a little bit, you want to give it an ice bath. Okay, one of the things that we're going to talk about later is the fact if we want to get the DNA out of here, we need this thing ice cold. So we're going to let it sit for about a minute. After that, you're going to smash it again. Again, we want to make sure we get all of those materials broken down. After that, another ice bath. And I believe the instructions say to repeat this even a third time. Okay? At this point, we're ready to take our banana soup here, and we're going to filter it out using a coffee filter and this funnel. And it should fit in there just fine. If you're having troubles getting it to stick, just drop a little bit of water in there and it will adhere to the sides of the funnel. Next, you'll need a test tube. This will be a lot easier if you had a partner. I do not have a partner tonight. We're going to dump our banana stuff in through the filter, and we're going to let it run into the test tube. You don't need all of it in there if it doesn't fit, just enough to get it working through. And it goes rather quickly. All we really need at this point is about 3 milliliters worth of solution, which is about half of the test tube full of this material. I'll hold it sideways there so you can see it's running and dripping in there. I'm going to stop a little bit early. I could let it go a little bit more. You want to take your banana stuff and the filter and throw that in the garbage. Also, rinse out and wash your funnel. At this point, we're going to grab our ethanol. Now, we got to be safe with ethanol. This stuff here is about 95% pure. And we want to take our eyedropper and we want to fill about 1 eyedropper full of the ice cold ethanol solution. And we're just going to dump that in with our banana stuff. And sure enough, there it forms. I don't know how clear it is for you guys, but the cloudy kind of gooey layer at the top that's forming right here is the actual DNA. And what happens with that ethanol being ice cold and the banana stuff being ice cold, it forms what's called a precipitate. That precipitate is actually the solid DNA from inside our cells. I hope I didn't make it look too easy, and I wish you guys the best of luck, and we'll chat on Monday how it went. Thank you.